Hello everyone, welcome to today's class. Today we are going to see class 12 biology chapter 1 sexual reproduction in flowering plants. In this today we are going to see the pre-fertilization and structures and events. Let's start with the topic children. So sexual reproduction in flowering plants begins long before fertilization occurs. Do you agree? Yes. So what is fertilization? The fusion of the male and the female gamete we call as fertilization. But the events that take place before the fusion of the male and the female gametes are called pre-fertilization events. So these events, it ensures that healthy male gametes are produced and successfully they reach the female reproductive part of the flower. So you have to know what are the major pre-fertilization events. They are like the first one is the formation of the pollen grains, nothing but our male gametes and development of the anther and the microsporangium and also microsporogenesis. What is that? That's the formation of microspores, development and maturation of pollen grains. So these processes, they occur inside the stamen. What is stamen? Is the male reproductive organ of a flower. For example, we can say in plants such as hibiscus, mustard, lily, and sunflower, pollen grains are produced inside the anther before pollination takes place. Okay, let's move on to the next one. Stamen, microsporangium, and microsporogenesis. Let's see the structure of a stamen. So what is a stamen? A stamen is the male reproductive organ of a flower and it consists of two parts. One is the filament and another one is the anther. What is this? The filament is a long, slender stalk. It supports the anther and helps position the anther for effective pollen release. And two parts it is having that distal part, it is attached to the anther lobe and its proximal part is attached to the, that is the ends of this filament. It is attached to the stalk of the flower, anther. So the terminal swollen structure we call as the anther lobes. Usually they are bilobed. They have two lobes. So each lobe contains two microsporangia. Therefore, a typical anther contains four microsporangia. So example, we can say the long stalk and yellow anther visible in a hibiscus flower. You could have seen no hibiscus flowers. Those are nothing but the parts of a stamen. Microsporangium, we can call that as a pollen factory. Why? Because it's a pollen producing structure present inside the anther. What is its function? It produces microspores, develops into pollen sacs and stores pollen grains before release. Let's see the structure of the microsporangium wall, children. Four layers are there, right? The first layer, you can able to see here, the first layer is the epidermis. Yes, this is the epidermis, first layer. Second layer is the endothecium. And the third one is the middle layer. And the fourth one, which is present inside, is the tapetum. Let's see it. So first one, epidermis, is the outermost layer. It protects the internal tissues. Next comes the endothecium. It helps in anther dehiscence. What is the dehiscence? Nothing but opening, right? And also it assists pollen release. Next comes the middle layer. It is located in between endothecium and tapetum. They are temporary layers and they provide protection during development. Tapetum. Tapetum is the innermost layer, children. They are rich in nutrients. That is why it supplies food to developing pollen grains. Here are some important NCERT point. Yeah, make a note of it. These tapetal cells often contain more than one nucleus. That is why they are multinucleate in condition, right? So I'll give you some frequently asked CBSE board exam question. Why is tapetum important? It's a 2-mark question. So answer is tapetum is the innermost nutritive layer of the microsporangium. What it does is supplies nutrients and grows substances which are necessary for the development of the pollen grains. Next, let's start with the sporogenous tissue. So where is this sporogenous tissue? It is present inside this tapetum. So at the center of the microsporangium lies the sporogenous tissue. What is its function? It gives rise to pollen mother cells. We call that as the PMCs in short. So it is responsible for producing the pollen grains. What is microsporogenesis, children? See, the process of formation of microspores from pollen mother cells through meiosis is called as microsporogenesis. Yes. So stepwise procedure given here. So first one, in the step one, what happens? Sporogenous tissues differentiates into PMCs, right? And then what happens? Each PMC is diploid. Diploid means 2n condition. And then in step 3, what happens? PMC undergoes meiosis. In step 4, 4 haploid microspores are formed. And in step 5, what happens? These microspores remain arranged in a tetrad. And at last, microspores, they separate and develop into pollen grain. Why meiosis is important, children? Because it reduces the chromosome number by half. That is why it is very important. For example, if PMC contains, let us, yeah, one example. If PMC contains 24 chromosomes, that is 2n, after meiosis, what happens? Each microspore will contain 12 chromosomes, that is 1n. This ensures chromosome number remains constant after fertilization. You have to draw and practice this diagram, that is TS of young anther, very important. You have to label like epidermis, endothelium, middle layers, tapetum, sporogenous tissue, very important. Always use pencil, children. Yes. And also label the parts on one side and underline the labels also. Let's go to the next one. So here we are going to see the pollen grain structure. What is a pollen grain? Pollen grain is the male gametophyte of the flowering plants. How they look like? They are microscopic. They are spherical. The size ranges from 25 to 50 micrometers, and they contain the male reproductive cells, and also they are two-layered wall. Yes. So let's see the structure of the pollen grain. The pollen wall, it consists of two layers. The outer layer is the exine and the inner layer is the intine. So exine is made up of sporopollenin. So what is its properties? They are extremely resistant. They are resistant to heat, they are resistant to acids and alkalis, and not degraded by enzymes. Importance: because of the sporopollenin only, these pollen grains can survive for thousands of years, like a fossils. Yes. So we can say like a fossil pollen grains. It helps the scientists to study the ancient vegetation and climate. Germ spores. What are these germ spores? See, certain regions in exine, they lack this sporopollenin. So these regions are called as germ spores. What is this function? See, during this germination, pollen tube emerges through germ spores during emerging germination. Intine. What is intine? Is the innermost layer. It is made up of cellulose and pectin. What is its function? It provides support and helps the pollen tube formation. Okay, the cells which are present in this mature pollen grain, one we call as the vegetative cell. What is this characteristics? They are large cell, they are rich in food reserves, and also they contain large nucleus. And what is its function? It forms pollen tube. Next is the generative cell. They are smaller size, dense cytoplasm, and spindle shaped. What it does? It produces two male gametes. So here are some pictures I have given for two-celled and three-celled. So in a two-celled pollen grain, we can able to see one vegetative cell and one generative cell. It occurs over 60% of the flowering plants. In three-celled pollen, you can able to see the generative cell divides by mitosis and it forms two male gametes. So it contains now vegetative cell, male gamete as well as another male gamete. So two male gametes, right? And for two-celled pollen, example is lily. For three-celled pollen, example is grasses, many grasses. That's a CBSE important question. Why is sporopollenin considered biologically important? It's a 3-mark question. You can write the points like they are most resistant organic material. Yeah, what is that? Sporopollenin. Yes, they are most resistant organic material. It protects pollen grains from environmental damage and also helps to preserve pollen fossils for millions of years. Next, let us see Microsporogenesis. So step by step we are going to see. The first one is the sporogenous tissues. It forms the pollen mother cell. Then meiosis undergoes and it forms the microspore tetrad. And at last the individual mature pollen grains are formed. Let's see this step by step. So in the first step what happens, this sporogenous tissue, homogeneous mass of cells are present in the center. What is homogeneous, children? Same cells. Homo means same. And then mass of cells are present at the center. Then what happens? PMC formation occur. So some cells become pollen mother cells. Next, this PMC undergoes meiotic division. And then the four haploid microscopes remain attached. Yes. And then the microscopes separate from the tetrad, and at last each microscope develops into a mature pollen grain. Let's see the significance of this microsporogenesis. First one is the genetic variation. So what happens? See, meiosis creates genetic diversity, right? So maintenance of chromosome number is very important. It ensures the correct chromosome number after fertilization, and also the production of the male gametes. It provides the pollen grains needed for reproduction. Let's see some application of sporopollenin. First, it is helpful in the fossil studies because ancient pollen grains, yes, they remain preserved because of sporopollenin. And then palynology is nothing but the study of the pollen grains, and plant identification. So defined exine patterns, they help to identify the species. And at last one is the allergy. When you take this parthenium, carrot gas, yes, it causes asthma, bronchitis, allergy reactions. CBSE frequently asked question is, what is palynology? So palynology is the scientific study of pollen grains and pores. Next, we are going to see the pollen, significance, viability, and pollen banks. Pollen allergy. Actually, many pollen grains, they trigger the allergic reactions. Symptoms are like you have sneezing, asthma, bronchitis, and eye irritation. Example like this parthenium pollen is highly allergenic. And pollen as food. So pollen grains are rich in proteins, vitamins, minerals, amino acids. It is it is available as tablets and health supplements and also nutritional syrups. Example like bee pollen supplements are marketed as health food in several countries. Next is the pollen viability. What is this pollen viability? The ability of the pollen grain to remain alive and capable of fertilization. What are the factors which affect this viability? Temperature, humidity, and plant species. For example, we can say when we take this rice and wheat, they remain viable for only about 30 minutes. You would have seen some food products, children. When you buy food products, you regularly used to see when is the manufacturing date and also the expiry date, right? The same thing happens here also. Viability is nothing but the manufacturing and its expiry. That mid interval is there, no? That one we called as the viability period. So for rice and wheat, it is just 30 minutes. For Rosaceae family and Leguminaceae and Solanaceae, they remain viable for months. Examples are apple, pear, peach, yes. Pollen banks. So what is a pollen bank? The facility where pollen grains are stored for long period. So what they do there, they store these one in liquid nitrogen at minus 196 degrees Celsius. This process we called as the cryopreservation. So liquid nitrogen is used, right, children? Yes, what is the temperature? Minus 196 degrees Celsius. This phenomenon we called as the cryopreservation. What is the purpose of this? It helps in plant breeding, hybridization, and the conservation of the genetic diversity. So scientists, they store this pollen, yes, so from disease-resistant crop varieties and use them later for breeding superior plants. Here are some NCERT important points. So the stored pollen remains viable for many years under cryopreservation. Let's move on to the next one. So let's see a quick revision, that is a complete summary of whatever we saw today. So stamen are the male reproductive organ consisting of filament and anther. Anther, they are bilobed, they are dithecous. It contains 4 microsporangia. Microsporangium, they produces the pollen grains. Tapetum is the innermost layer, nutritive layer, and they are multinucleate. PMCs are pollen mother cells. They are 2N, that is diploid, S, and microsporogenesis. So pollen mother cell undergoes meiosis. It forms microspore tetrad and it gives rise to pollen grain. Pollen wall contains two walls. Outer is the exine, which has the sporopollenin, is a resistant layer, and intine is the inner layer composed of cellulose and pectin. Germ pores is a sites through which pollen tube emerges. Pollen viability. So it depends upon the temperature, humidity, and species. So pollen banks, they are stored in liquid nitrogen at minus 196 degrees Celsius for future crop improvement. So let us have some 1 mark CBSE questions for revision children. What is a microsporangium? A pollen producing structure present in the anther. Which layer nourishes developing pollen grains? Tapetum. What is sporopollenin? The resistant material present in exile. What is a tetrad? A group of 4 haploid microspores formed after meiosis. What is microsporogenesis? Formation of microspores from PMCs through meiosis. At what temperatures are pollen grains stored in pollen banks? Minus 196 degrees Celsius. What are germ pores? Regions of exine lacking sporopollenin through which pollen tubes emerge. Name the two cells of a mature pollen grain, vegetative cell and generative cell. What is palynology? Study of the pollen grains and spores. Which cell produces male gametes? Generative cell. As for CBSE board exams, you please focus on structure of anther and microsporangium, and also tapetum functions and microsporogenesis flowchart, and also pollen grain structure diagram. That is very important, children. Just practice those diagrams, and sporopollenin properties, and also pollen viability and pollen banks. So we have come to the conclusion of this part. We'll see you in the next video. Yes, watch all the videos and don't forget to subscribe. Thank you.